Basile Tarchini is an associate professor at the Jackson Laboratory, and he's a research associate professor at Tufts University School of Medicine. His BS in biology is from the University of Geneva in Switzerland, as well as his PhD, and then he realized that he should be in Maine. I'm adding that part. You know, to that part. His primary scientific interest is understanding how genes and proteins exquisitely orchestrate the development of cells and organs across the body. His doctoral research in Switzerland addressed how a family of tightly clustered genes organized neurons in the spinal cord and also formed distinct digit in the limb. During his postdoctoral research in Canada, he studied the architecture of sensory hair cells in the inner ear, identifying proteins that define the placement and elongation of stereocilia, the motion sensors that are essential for hearing. And I will say that was what caught my eye about your research, because I don't know of a lot of people doing that kind of inner ear research, so I thought it was really cool. So these projects laid the foundation for his independent research at JAX, where he studies inner ear development, how sensory cells in the inner ear acquire the ability to detect sound, gravity, or head movements. Their research informs emerging therapies aimed at repairing sensory cells damaged during life. And then I'm going to read your whole thing that you wrote about this because it's great. Oh. Basile's pop culture is the movie Alien from 1979. I'm reading this because if this inner ear research doesn't work, he can go into film critique. A masterpiece. It triggers curiosity about alien biology, how their larval stage uses humans as hosts to develop to adults, great storytelling uniting science, technology, psychology, and even humor, unique pacing and aesthetics. And I could not agree more. It's excellent.
Okay, thank you for having me. So I'll start with the basic. I mean, sound is really air vibrations that can travel down your ear canal and then set in motion those small bones in the middle ear. And then they're pushing and moving fluid in the inner ear. The inner ear is really just one of the bones in your skull, and it has a dorsal side, with vestibular organs that are responsible for the sense of balance. And it has a ventral organ that is really the cochlea, the auditory organ. So we cannot crack open a human cochlea to study it. That's what we're using. That's why we're using animal models. The mouse, the mouse in our case. And so why? Because the mouse has a similar set of genes and architecture of genes as humans, has a similar inner ear anatomy and physiology. So it's a great model to study how the inner ear normally works, but also to model genetic diseases and find therapies. So in the mouse, if we look at the cochlea, we see that the sensory region is this thin coiled region here that is along the organ, and sensory cells detect different frequencies along the coil, a bit like a reversed piano, if you want. If we zoom in further, we see the surface of individual sensory cells. So that's one of them here. So we call them hair cells because they have this bushy top, and I'll get back to that in a second. And what's really very important to keep in mind is that hair cells, once injured, do not regenerate. So once dead, never comes back. You are born with a set of sensory cells that you will use for your whole life. So we are very interested in this surface structure, the bushy top, what we call the stereocilia bundle. This is really the motion sensor of the cell. So sound is deflecting those structures, activating the hair cell, and then this hair cell transmits the signal to neurons that transmit, in turn, the signal to the brain. In my lab, we're really interested in how the motion sensor gets sculpted during development by architect proteins like this protein GPSM2 in red. You see in a normal cell on the right, the protein is sitting at the tip of the stereocilia, and that's why they get really tall, leading to normal hearing. But in a mouse model that is lacking that protein, no red signal at the surface here, you see that the stereocilia get very stunted, very short, leading to profound deafness, both in this mouse model, but also in human families that have similar mutations in this gene. So hearing loss is really has multiple causes, and it's one of the most frequent birth defects, one of the most common health issues, and actually it's a health issue that is on the rise because we live longer lives. Yet, as I mentioned, sensory cells do not regenerate. So hearing loss becomes progressively a bigger burden. Hearing loss is not just hearing loss. It's also being more prone to accidents. It's also, at older ages, social isolation and depression. And it's been associated with cognitive decline, especially Alzheimer's disease. So what do we do about it? I mean, until recently, all solutions revolved around engineering. So this type of funky contraption from the past has been translated today into hearing aids, which are very simple but also very useful amplifiers for low sounds. For profound hearing loss, we use surgical solutions where a cochlear implant, which is really an array of electrodes, is implanted into the ear and can substitute to the missing or the damaged sensory cells and stimulate the nerve. But what's really exciting is that only in the last couple of months, a new solution, a biological solution this time, has been put forward. And this is in the form of a first successful gene therapy for a protein called otoferlin that sits here at the interface between the sensory cells and the neurons, and that is missing in some families. And the idea is to bring back that protein. So this little movie is taken from a publication from January this year. And you see, if we can play it, yeah, that this kid, I guess there's no sound, but this kid cannot hear his father call his name because he's completely deaf. And here, only 6 weeks after injecting a normal copy of this otoferlin protein, this kid is able to hear his father call his name in his back. And other kids in those trials are starting to speak, which is absolutely incredible. And at this point, I'd just like to highlight, and I think it's really important, that this has only been made possible by decades of research using animal models to make this procedure very effective and also very safe. Thank you so much.
I've read that birds can regrow the hair cells when they're lost.
Yeah, that's correct.
Why do you think it is that mammals can't do that?
So if we're talking about the auditory cells, the structure of the auditory organ is pretty different in mammals. It's much more specialized and complicated, and we think that the cellular architecture in the auditory organ just makes things really difficult for new hair cells to be produced from progenitors. So it just doesn't seem to happen. It does happen a little bit in mammals, but in the vestibular system, the sense organs, the twin organ in the inner ear, but not in the auditory system. And this really, I mean, in the end, this is really maybe one of the biggest take-home messages, that those cells are so specialized that we can't really, I mean, there's a lot of research being done to try to regenerate them, but so far it's not something that's been really done successfully. And so it's really worth thinking about that. I mean, each time you listen to loud noise or something, I mean, it's a few cells that go away, they don't come back, and just piling up with age, literally.
That's a great lead into the question I had. So the study that you quoted of the child that could hear, that was basically re-injecting through a viral insert, that they reinserted the detection protein.
Yeah, yes, that's a great point. The protein, the gene for that protein is mutated in that family, and what they're doing is using a viral vector to bring back a normal functional copy of that protein into her cells.
Right. So I'd like you to comment on when you have structural damage, like what you've been talking about, right? So which is a different issue. Yeah. Can you comment on how to use gene therapy to repair structural damage that's been generated?
So that's a great point. I mean, this is already really a technical point, but it's very important. There's 5 or 6 different consortiums that are doing the same gene therapy trials on this otoferlin protein. And the reason they all chose the same protein is because there's no structural damage when you're missing that protein. It's just like a transmission protein, if you want. And the problem being that many other mutations are actually damaging the hair cell. So if you introduce that protein, often it's too late. The damage is done, and the cell, as a little factory, is not functional anymore. It's not enough to bring back that protein. In a way, it's too late. So a lot of those gene therapy trials, the concept is really to hit the right time window where it can be useful before it's too late.
Could you talk a little bit about tinnitus and whether what you're doing can relate to that, and if there's anything that's in the
Yeah.
works for curing?
So it's really not my specialty, but so tinnitus is really a mystery. What I can tell you is that it seems to have very little to do with the inner ear, actually. So think of the inner ear as the peripheral organ that is dealing with sensory perception, and tinnitus seems to be born in the brain. So those sensory signals are relayed to the hindbrain, and then they're moving up to the cortex. And it's at the level of this further processing that